To our next talk by GEMMA CARVILL from Northwestern University, who's part of the NIH-funded Center Without Walls, EPI-MVP, and the goal there is exactly to do that, to integrate functional assays and use those functional assays to improve machine learning and prediction of pathogenicity. So we're going to hear from her about SYNGAP. Did you take the clicker? No clicker. I'm going to click my fingers and the slides are going to change. No? Oh, wait, is it this?
That's it.
Oh.
See the green arrow?
I thought it was a mic. Well, that's a good start. Thank you, Heather, for the kind introduction. I'm GEMMA CARVILL. I'm an assistant professor. I'm from Northwestern University. I'm really presenting here on behalf of the entire EPI-MVP. So this is a large collaborative project from multiple different institutions. I'm going to chat about it a little bit more in detail, tell you what we're doing. If I stand over here, can you all still hear me? I feel like there's a lot of interference there. Great, awesome. Maybe. Promise I'm pressing the green button. Oh. Fabulous, great. So we've had two fabulous introductions of variants of uncertain significance, which is the focus of our work as well. And this, as has been described, are really those variants where we don't have enough information to make a call one way or another as to whether that particular variant is associated with disease. And these, of course, are differentiated from likely pathogenic and pathogenic variants, which are highly likely to be disease-causing, versus benign or likely benign variants, which are unlikely to be related to the disease that we're studying. And so VUSs are a geneticist's nightmare slash cash cow, I guess, because they'll keep us employed for at least the next 20 years, I think. And this is really illustrated here in this slide. So what I'm showing you is the classification of all variants in a large database, which is called ClinVar, which is essentially a collection of variants that were detected via genetic testing. And so this is just in the last 2015 to 2020. And so what you can see is that in gray there, VUS really make up the biggest percentage of results from clinical genetic tests. And the other really worrying thing is if you look at the graph on the, I guess, left, right there, you can see that this VUS number is really increasing almost exponentially. And so this is a huge challenge for geneticists, and I think we need a lot of different approaches to try and tackle these VUSs. And so ClinVar, of course, collects information from a variety of different disorders, but just zoning in on epilepsy-related VUSs, and here I'm really focusing on missense VUS. And so I'm showing you here the data from 4 epilepsy-related genes. These are probably the most prominent non-ion channel epilepsy-related genes. And what you can immediately appreciate is the PLP variants, so pathogenic, likely pathogenic from now on, I'm just going to call PLP. The benign, likely benign variants, I'm just going to call BLB from now on. And then, of course, yellow is VUS. I recognize it's now a little confusing because I switched colors from Dr. Courtney. Our rationale is that epilepsy's color is purple, and so the pathogenic ones are purple. Gray we don't care about, and yellow is like attention, we need to figure these guys out. So that's our rationale for the color scheme. And so what you can immediately appreciate from this slide is that missense VUS really represents the largest category of variants, and we really need to be able to have better tools to establish which of these are pathogenic and which are benign. Again, because this limits access to clinical trials, which we've heard a lot about today. An individual has a missense VUS, they are excluded from those trials, and also often from further genetic testing, whether that's pursued is also a somewhat nebulous outcome as well. And so you can see there are many hundreds of VUSs. And so probably about 5 years ago now, we wrote a grant for a big U54, so this is a large Center Without Walls, that was eventually funded. We call it EPI-MVP. The MVP is a well-known sports term that none of the reviewers picked on that I thought was very clever, but it also stands for multi-platform variant prediction. So EPI-MVP is led by PIs Laurie Isom and Jack Parent at the University of Michigan. We're involved there from the Gene Variant and Curation Core, and so we're really the genomics and computational hub of the project. And then there are 3 projects that I'm going to talk about in a little bit more detail a little further along. And as I mentioned, this is a US-wide collaboration between Weill Cornell, University of Michigan, UCSF, and us at Northwestern, and Heather at St. Jude as well. And so I showed you this previous slide with those 4 genes for a specific reason, because those are the 4 genes that we're studying in EPI-MVP. And so SDXBP1, PCDH19, SYNGAP, and SLC6A1. We're furthest along, really, with SDXBP1 and PCDH19, and so much of what I'm going to present to you today on SYNGAP1 is mostly the computational side, but I'm also presenting here as a call for collaboration. We would really like to collaborate broadly with anybody doing protein modeling, for instance, doing high-throughput screens, anybody who has ideas about which functional assays we should be using. And so really how EPI-MVP works is our goal is to integrate computational and functional data sets to resolve variants of uncertain significance in the epilepsy-related genes. And so our goal at GVCC is to build a machine learning model where you can give the model a particular variant of uncertain significance, and it will then predict what is the likelihood that that variant is pathogenic versus benign. And so to do this, what we do is take known pathogenic and benign variants in a particular gene, and we train the model. And I'll go into that in a little bit more detail in a moment. And we then give it VUS, and we score those VUSs. And then the beauty of this particular setup is that we then can test the power of the accuracy of our predictions in several different cellular systems and animal systems. So we have primary cells and eye neurons, and that's head up by Betsy Ross at Weill Cornell. We have 2D and 3D iPSC, so both patient and CRISPR-generated SYNGAP1 and other models, as well as animal models, so primarily mice, but also a little bit of fish as well. And so the idea then is to test our predictions in those animal model systems and re-inform the prediction algorithm. So as I mentioned, we're furthest along in terms of the computational modeling. And so to do that, what we first need is to get a good sense for the landscape of missense variants in this particular gene. And so we spent a long time collating missense variants. This data comes from ClinVar, it comes from our industry partners, GeneDx and Vitae. It comes from collaboration with SYNGAP, with SRF, for instance, with their known pathogenic variants literature, and a variety of different sources. NOMAD is another one that we use extensively for the benign and likely benign variants. And so all of the data today is going to be presented from that. But we started integrating new data sets as well, which I'm happy to talk about at the end in relation to a question that was asked earlier. And so what we can do then is look at the distribution of these missense variants across the gene. And so again, the PLP variants, there are 51 of them, they're there in purple. And as was shown earlier, they really tend to cluster in the PHC2 and GAP domain, with only 4 pathogenic missense variants in the DUF domain. Conversely, if you look at the benign and likely benign variants, so there are about 59 of them, they tend to cluster away and more towards the disordered domain. And then, to the surprise of absolutely no one, the biggest category are the VUSs, and there's about 241 of them, and they are broadly distributed across the protein. And so with these 59 BLB and 51 PLP variants, we can then go and train the model to distinguish between pathogenic and benign missense variants. And so we start with those variants, and then we do feature annotation. And so in the current beta version that I'm presenting today, we have about 30 different features that we use to annotate each of those variants. And so we're using things like conservation, we're using amino acid properties, we're using protein structure, stability, surface change, a variety of different metrics. And for every single missense variant, we're ascribing a score to that variant. We then use fairly simple machine learning approaches, so supervised approaches such as random forests and decision trees, to then train the classifier to be able to distinguish between pathogenic and benign variants. And so when we do this, in reality, what we do is we split that data set of about 100 variants into a training set and a test set. And so the training set represents about 60% of the data, and we train the model to distinguish between pathogenic and benign variants. And so doing that, we can do that with about 99% accuracy. And then we hold out 40% of the data set because the model has not seen that, and then ask the classifier to assign a category and ask how accurate is that. And so there we get it about 88%. And so another way to visualize this is with this confusion matrix here. So basically the two dark blue sections are where the model accurately classify a variant. And the two lighter colors, so where you see 1 and 1, are our false negatives at the bottom and our false positives at the top. And so you can see the training set there, you can see the test set next to it, so it gets 3 and 3 wrong. And so we're maybe a little bit overfitting here, and some of this is just really the challenge of this being a pretty small data set. 60-ish variants in each class is actually pretty small. And so we have some ideas on how to make that bigger and better, and I'll talk about that more towards the end as well. And again, very open to folks' ideas in that as well. So the other thing we like to do is benchmark against the tools that are out there. So CAD is one of the tools that many people use to determine, or to not really determine, but get a sense for whether a variant is likely to be pathogenic or not. And so if we compare to CAD, we perform slightly better than CAD using this gene-specific predictor. So the difference between CAD is CAD can generalize across the entire genome, whereas we only care about one gene. And so our model is not generalizable to the genome, but we don't care because we just care about SYNGAP1. And so the other thing that we can then do, now that we're fairly confident in our model, is that we can generate a score. So we call this the probability of pathogenicity score, or the Syngap PPLP score, because we like to say hard things like PPLP in talks for funsies. But what I'm showing you in this graph is you can see there the scale goes from 0 to 1. 1 would be a variance that is highly likely to be pathogenic, scored by the model, whereas 0, the model thinks that that variant is highly unlikely to be pathogenic, so BLB. And so you can see, as one would expect, that the BLB variants tend to cluster towards 0, whereas the PLP variants tend to have scores towards 1, as one would expect. We can then score all of those 241 VUS and ask where do they lie in terms of the prediction. And so if we set a threshold of about 0.5, which is what allows us to most accurately discriminate between BLB and PLP variants, although it is a little bit arbitrary and we're working on more sophisticated methods. But if we do that and draw that 0.5 as our threshold, we then take away that about 22% of those VUSs, or about 54 missense VUSs, are suggestive they could be pathogenic. And so this is somewhat encouraging because this may be the diagnosis for almost a quarter of patients. But of course, this is just a model, and so we need to validate this model. The other thing that I was always concerned about with making a machine learning model for SYNGAP is that I showed you in the beginning how where that missense variant is really matters in terms of whether it's likely to be in the general population or pathogenic. And so one of the things that we first did was ask, well, if we look at the distribution of scores of the VUSs and our PLP variants in relation to where they sit in the genome, is it purely just amino acid position that's driving our model? And so what I'm showing you here, again, is the PPLP score on the y-axis, left, right, x, y, on the y-axis. So at the top are the variants that are likely, that the model thinks are likely to be pathogenic. 0 are those that are likely to be benign. And so you can see there in purple, particularly in the PHCA2 and GAP domain, they tend to have very high scores. In the DUF disorder domain, there is definitely a bias, but it's not as bad as I had originally thought, in that you can see that there are those 4 PLP dots, and at least 3 of them are above the threshold for pathogenicity, and then some missense variants as well. And so this is a nice visual way of looking at how much amino acid position drives the classifier, but there are also more sophisticated ways to look at this. So this is essentially a SHAP value, a SHAP model. And so what we're doing here is basically asking, of those 30 features that we gave the machine learning classifier, which ones are being used, and which ones are being used to drive the model most predominantly. And so you can see the 4 at the top there are really based on conservation, and there is certainly, I don't have a pointer, but AA-pos basically means the amino acid position. So the amino acid position is definitely being taken into account, but it's not completely driving the model. The other nice thing that SHAP can give us is, beyond the conservation, what are the other features that drive the model? And so one of the things that we noticed was that a missense variants that change polarity as well as charge was something that drove the model. And changes in polarity and charge can often affect stability and aggregation of the protein. And so this is driven purely computationally, and so I've been very excited to see some of the talks this morning where folks have shown both in neurons, and, some cell models as well, that some of these missense variants can cause aggregation. So I think it's kind of neat that our computational side and the functional side, working completely independently, might be telling us the same thing. And so, and again, that was not a plant. I am not a plant. I did not know about that before this. There is one paper that shows that some missense variants can aggregate, but N of 1. So, to my knowledge anyway. So what do we do next with this information? How can these models inform how we interpret variants? And so our next goal within EpiMVP is to do functional testing. And so we have 3 PLP and 3 BLB variants. They are basically our positive and negative controls. And then we selected 4 VUS for functional testing. We select VUS across the range of PPLP scores because I think it gives us a more accurate reflection of how good our model is. In an ideal world, I'd like to take those 54 variants that are all PLP and run them through a couple of models and see if we can really reclassify them. And so that's a story for another day, but this is the approach that we're taking right now. And so what these variants will now have, they've actually all been made already and have moved at least into Project 1, which is in primary cell lines. So I think we're using HEKs and another obscure one that I can't remember. And the goal is to do overexpression and test things like stability, aggregation, also phosphorylation of some downstream targets. And then we're also doing iPSCs and mice where we have knockout models, and then we're trying to rescue phenotypes both with wild type but also with the constructs that contain the missense variants to see if we can rescue those phenotypes. And again, feed this back into the accuracy of the model. And then one more thing, EpiMVP adjacent. This is great for doing kind of a couple of variants at a time, but we're also very interested in my lab at generating hundreds to thousands of endogenous edits. And so again, this is editing the SYNGAP1 locus rather than doing overexpression and developing high throughput readouts so that we can combine that with the computational analysis as well. The very last thing I will say is that this also comes to an earlier question. Our goal is we are building EpiPred. EpiPred for STXBP1 is nearly ready. The idea is that anybody in the community, scientist, family, clinician, will go to the website, enter in your missense variant, and what you'll get is the score. So whether we think that variant is pathogenic or not, as well as a big explainer on how we arrived at that score, so what's driving that. And so I will end there because I see Heather hovering. But just to say, I would love to talk to everybody at the break in terms of functional models, but also for EpiPred, what you would like to see as families and clinicians and scientists. And also come collaborate with us, we're awesome, because here's the team.